time. So welcome back. Welcome to also to anybody who wasn't here yesterday. We had a great day yesterday. We're going to start this morning with a science session. So I'm going to turn it over to Amy Arnsten to introduce the first session.
Thank you so much. Good morning, afternoon, wherever you are. And we're now going to have our several 3-minute talks. And if you could please enter your questions in the chat, and I will try and capture them. And then when our talks are over, I'll call on people to ask their questions out loud so that we can then have a discussion. Let's begin. Good morning. I'm Amy Arnsten from Yale Medical School, and I'd like to introduce you to our new NeuroNex on the fabric of the primate neocortex and the origin of mental representations. Our group hopes to learn how cortical circuits generate the mental representations that are the foundation of abstract thought. Early research found neurons in the rhesus monkey dorsolateral prefrontal cortex that can generate mental representations of visual space as the monkeys performed a spatial working memory task. These so-called delay cells exhibit spatially tuned persistent firing that is maintained across the delay period, even without sensory stimulation. Parallel in vivo and in vitro studies by Goldman-Rakish and Gonzalez-Burgos showed that this persistent firing arises from extensive recurrent excitation between pyramidal cells with shared characteristics, while the spatial tuning is refined by lateral inhibition from PV-containing GABA interneurons. The research in our NeuroNex will build on this initial scaffold to learn more about the neural basis of persistent firing, including the role of NMDA receptors mediating the recurrent excitatory connections, and the multiple roles of interneurons, such as potential disinhibition of persistent firing by calretinin-containing interneurons, which the Martinez lab has found enriched in dorsolateral prefrontal, as predicted by Xiao-Jing and Wang's computational models. We'll also explore the molecular characteristics that allow persistent firing, including transcriptomic analyses across cortical regions and across species, for example, as just published by the McGreal group at Harvard. And we'll be looking into evidence of magnified calcium signaling in the dendritic spines of these circuits compared to those in classic circuits. This will include evidence of calcium magnification from immuno-EM, which may serve as an important bridge to the new NeuroNex on synaptic weights. We'll explore these hypotheses across cortical regions and across species, especially comparing the marmoset to the rhesus monkey. We have 4 IRGs. IRG1 examines persistent firing in rhesus and marmoset in vivo. IRG2 examines identified neurons in vitro. IRG3 examines molecular differences across cortical areas and species. And IRG4 will integrate these data using computational modeling. The work involves 15 labs in 3 countries, and we will hear from Julio, Guillermo, and Xiao-Jing in the upcoming talks to say a bit more about our approach. Thank you.
Hello, everyone. I'm going to tell you about what we are going to do in the IRG1 of our NeuroNex, and Amy already introduced. We are 4 investigators from Canada, the United States, and Germany. Working memory is the maintenance and manipulation of information in the mind for short time periods. It is a type of mental representation. The neural correlate of working memory is the persistent firing of neurons representing the memorized item. In this case, these neurons recorded from the macaque prefrontal cortex represent a remembered location out of these 8 different locations. And it was first described in 1971, and it has been corroborated by different studies. The question that we asked in the past is whether all brain areas have the ability to produce persistent firing encoding working memory. In this study, we recorded from areas MT, early visual area, for area MST, early association area, and for the lateral prefrontal cortex, late association, or executive cortex. We found that working memory was encoded by neurons in areas MST and the lateral prefrontal cortex, but not an area MT. What you can see here is just the firing of several example neurons. In this part of the where the animal is remembered the direction of the stimulus, these neurons are firing here, representing the contents of working memory, but not in area MT. So the idea here is whether are there properties of neurons or circuits in association areas that are essential for the encoding of working memory, and are those universal across anthropoid primates or perhaps other species? This is, in a nutshell, the main question of our NeuroNex. In IRG1, we are going to record the responses of neurons in 3 different areas along the hierarchy of visual processing that have homologs in macaques and marmoset monkeys. And we are going to use neuropixel probes. The areas are V1, LIP, and LPFC, and these are the 2 species that we were going to record from. We will probe the areas with a naturalistic task. Basically, we're going to take advantage of the animal's ability to navigate virtual environments. This is a macaque monkey navigating a virtual environment, doing a match-to-sample task. And I'm showing you one of the tasks that we intend to use. This is the macaque in a virtual arena, so the animal cannot move when this is shown in the arena. So basically, the task for the animal is remember the location of that red object and now navigate toward that location. You can analyze the data in different periods of the task, cue, delay, and response. And what we see in this particular neuron is that the neuron encodes working memory during the delay period of the task. Here, the different locations, and here, the firing rate in this color scale. So in conclusion, what our IRG1 is going to do is to sample our box, our NeuroNex box, at this specific level, at the level of circuit motifs, single neurons, and neurons. So, and hopefully we're going to fill the box with data from the other IRGs. Thank you very much. This is actually our team that you probably recognize from Amy's talk before. Thank you.
In this talk, I will share with you some published data from a study addressing one of the central hypotheses of our NeuroNex network, that gradients of cellular properties across areas, cortical areas of the primate brain are crucial for the neural basis of mental representations. Our study addressed the transcriptome physiology and morphology of layer 3 pyramidal cells, because this layer is thought to mediate the recurrent excitation necessary for persistent activity in working memory. Moreover, layer 3 pyramidal cells are thought to be crucial for cortico-cortical communication. To assess transcriptome, we microdissected layer 3 pyramidal cells from prefrontal and parietal areas of the macaque monkey neocortex and used DNA microarrays and quantitative PCR. And our studies actually reveal significant gradients of gene expression because more than 600 genes were differentially expressed by either prefrontal or parietal neurons. Moreover, some of our findings allowed us to predict some differences in the physiology and morphology of layer 3 pyramidal cells. For example, this gene, which is highly expressed by prefrontal neurons relative to parietal cells, encodes a calcium channel involved in bursting, therefore suggesting that perhaps prefrontal neurons show stronger bursting physiology. On the other hand, this other gene MET, which is highly expressed by parietal cells, encodes a negative regulator of dendritic tree size, suggesting that prefrontal neurons possess larger dendrites. In order to test these predictions, we use patch clamp electrophysiological recordings and self-filling in acute brain slices. And our findings were actually consistent with our predictions because, for example, we found that in the prefrontal area, layer 3 pyramidal cells with bursting properties were much more abundant. In addition, our morphological findings were also consistent with our predictions because we found that the basal dendrites of prefrontal cortex layer 3 pyramidal cells had greater total length and also possess higher density of dendritic spines, which are the main sites of excitatory synaptic input. And while our results have a number of functional implications that would be interesting to discuss at the end of this session, We think that this is just a very preliminary and limited view of the neuronal heterogeneity and gradients that exist in the cortex of primates. Therefore, in our NeuroNex network, we plan to implement a pipeline to collect patch-seq data in order to be able to much better characterize the neuronal heterogeneity and gradients, and therefore develop much better models of the functional implications of these gradients. Thanks for your attention.
I'm Xiao-Jing Wang, speaking on behalf of the International Research Group 4 that consists of myself, John Murray at Yale, Lyle Miller at Western University in Canada, and Fred Wolf at Göttingen University in Germany. We developed theory in close collaboration with our experimental colleagues, trying to achieve understanding across levels, from genes to neural circuits to function. Among projects and ideas we are about to explore, I'm going to highlight one hypothesis, namely that the cortex is organized according to a canonical circuit principle applicable to all areas. At the same time, biological properties change from area to area that can explain the distinct pattern of distributed working memory. What do I mean by that? So, for example, theory and experiments indicated that the NR2B subunit containing NMDA receptors are important for excitatory reverberation underlying working memory. Now there's evidence that the expression level of the gene that encodes NR2B shows an increasing hierarchy gradient along the hierarchy in the cortex. Another example is the spine count, which is the proxy of the strength of excitation per in the neuron. It also increases along the cortical hierarchy for macaque on the left and the marmoset on the right. On the other hand, in terms of inhibition, this proportion of carbandin or somatostatin positive interneurons versus the parvalbumin positive output controlling interneurons varies from area to area according to a macroscopic gradient. So the plan is to incorporate those kind of gradients measured by genomics, anatomy, and physiology into a large-scale cortical model for marmoset and separately for macaque to explore distributed working memory representation and the underlying mathematical mechanism as well as biological mechanism. So we're very excited to participate in this collaboration and working together with everyone else in this consortium. Thank you.
Hello, everyone. Today, I will present a research progress update from the Washington University and University of Illinois Urbana-Champaign NeuroNex Hub. The primary goal of our hub is to create tools and protocols for in vivo imaging of neuronal activity in diverse non-model organisms. In particular, we have focused on social insects, honeybees, and locusts. So today, I will be talking about the imaging pipeline that we have been working on. So this involves collecting volumetric signals using light sheet microscopy. So we have refined our approach using the fruit flies, Drosophila melanogaster, as a model organism. We are focused on the olfactory system and looking at 3 distinct neural populations: the olfactory receptor neurons and the 2 downstream targets, which are the cholinergic EPNs and the GABAergic IPNs. So let me now show you a quick video of what kinds of signals we have acquired from these 3 neural populations. What you will see is that there are 4 segments to this video. The first segment will present the calcium signals acquired from the receptor neuron axonal terminals entering the antennal lobe. There are 2 clips showing responses to 2 different odorants, and there are 3 columns showing responses collected from 3 different depths. So the second segment is going to show a similar film by showing responses, calcium signals acquired from the EPN dendrites, which is the top row, and EPN axons entering lateral horn, which is the bottom row. So here again, there are 2 clips showing the responses to 2 different odorants. The third segment shows a very similar movie showing the axonal activity of IPNs, which provide feed-forward inhibition to the lateral horn from the antennal lobe. So the last segment is going to show how we extracted ROIs from these movies using a non-negative matrix factorization approach. The individual ROIs corresponded to the individual glomeruli in the antenna lobe and axonal boutons in the lateral horn. We were able to acquire images at 4 Hertz from each optical plane, which basically means we had good spatial and temporal resolution to answer what were the inputs coming into the antenna lobe and how they were transformed into spatial temporal responses in the 2 distinct neural populations in the antenna lobe. So using this approach, we were able to acquire a lot of data, good spatial and temporal coverage, which allowed us to understand what were the spatial temporal response features of the 3 distinct neural populations that we examined, the ORNs and the 2 downstream targets, EPNs and IPNs. So in addition, we were also able to characterize the tuning of individual axonal boutons to different odorants in different flies and different lines. And this allowed us to compare how the feed-forward excitation and inhibition converged onto the lateral horn, whether they matched or mismatched, which is shown here using a color code. Similar colors means matching tuning, dissimilar color means mismatched tuning. For more information, I'm going to refer you to this bioRxiv paper that will appear later this week. Future work will look at transgenic comparative studies using transgenic locusts and honeybees and other insects. So with that, let me thank you for your attention.
In this presentation, I'd like to introduce high-density micro-LED optoelectrode developed at MINT-Hub at University of Michigan. And MINT-Hub at University of Michigan has been developing and disseminating a collection of tools that collectively enabling structural and functional mapping. And in this talk, I will introduce one of the tools that we are developing in this functional tool, optoelectrode. People used to use a fiber for optogenetic studies. A fiber is a bulky and rigid, and it has a limit in the stimulation site. So we directly integrate small micro-LEDs onto the Michigan probe shank. This micro-LED is so small, with neuronal size of about 10 micron. We can precisely locate it in the vicinity of the recording channel for cellular optical stimulation capability. And this prototype has a 32 recording site and also 12 micro-LEDs in 4-shank configuration. And we implant this probe into the hippocampus and stimulating pyramidal neurons selectively, which is 50 microns apart, by illuminating light into the 8 adjacent micro-LEDs and switching back and forth, which is in 60 micron separation. And currently we are developing high-density micro-LED, scaling the number of recording site and the illumination site. We are integrating more than 100 micro-LEDs into the probe shank, and it is not trivial to control precisely all these micro-LEDs. So we developed the control driver chips to driving this high-density micro-LEDs. And also, it is very important to reduce the simulation artifact in the high-density probe, because in this configuration, we have a recording and stimulation site on the same location, a very close location. By introducing shielding layers, multiple shielding layers, we could eliminate this artifact almost zero. So we could even record the spike signals during the transition of the optical stimulation. And this high-density probe has a prototype device currently, has 256 recording sites and 120 micro-LEDs, and it's now being tested in the in vivo animal, and this probe will be available to the user community sometime next year. So contact us if you are interested in these probes, getting some samples, and currently we are getting your survey and feedback. Finally, I'd like to acknowledge all my collaborators and researchers who developed this technology. Thank you very much.
Hello, I'm Ryan McGreal. I'll be giving the update for the next generation Multiphoton Neuroengineering Consortium, or mnemonic. Before we begin, I'd like to thank the organizers for the work and planning that they put into making this meeting happen. The mnemonic team is made up of over 40 members. A series of case studies for enabling multiphoton instrumentation are in progress. Christina Nielsen leads the study on chronic 2-photon imaging in larger animals. Prakash Kara leads the study on 3-photon imaging of calcium and glutamate in cats. Michael Gord leads the study on automated behavior rigs for training mice. Jerry Chin leads the study on advanced multi-area two-photon microscope, that is, exploring the limits of temporally multiplexed beams. And Garrett Stuber leads the study on imaging a cortical area simultaneously with a subcortical brain region through a grid lens. To advance fundamental technology, John Bowers and Luke Thielgarajan are developing new technology to enable untethered multiphoton imaging and stimulation in awake animals. Jeff Squire is developing imaging modalities that are capable of enhanced resolution. In addition, the consortium's lead PI, Spencer Smith, hosts a series of high-level technical meetings on emerging technology. In order to disseminate our findings, a series of workshops are held at the Prime Institute, UCSB. We also share all resources to not only disseminate, but also sustain technology developed here. Chae-Hong Yu is the project lead engineer, and I, Ryan McGreal, am the project manager. Our hub utilizes several custom-made microscopes spread across several sites. To the right is a video of our Diesel 2P scope. Diesel 2P, or Dual Independent Enhanced Scan Engines Large Field Two-Photon Microscopy, is located in Spencer Smith's lab at UCSB. Key features include a 5 by 5 millimeter field of view, and it can simultaneously and independently scan two regions. I have also included a link to the Diesel 2P preprint on the slide. In addition to Diesel 2P, there's the Quadrascope, a 4-channel two-photon microscope in Jerry Chin's lab at Boston University. Our next generation 3-photon microscope, Diesel 3P, has been designed and is currently being manufactured. Spencer Smith and Chae-Hong Yu led the design process, and the microscope will be assembled in Prakash Kar's lab at University of Minnesota. Briefly, I have a few images to share. Image 1 shows some 3-photon images taken in Prakash Kar's lab from his recent paper, and these are taken at 1300 nanometers. Image 2 is a black box design for an extra long working distance air objective, a 22 millimeter working distance, for 2-photon imaging designed by Chae-Hong Yu and Spencer Smith. We're finalizing quotes to manufacture this objective right now. The 22 millimeter objective is air immersive and for use in animals with thick cranial window preparations such as cats, ferrets, and non-human primates. If you have any interest in it, please contact me. My email is on the next slide. I'd like to end this by thanking the NSF, our funding agency, as well as all the participants in our hub. Thank you.
Hi, my name is Agostina Palmigiano.
My name is SueYeon Chung, and we work at the Center for Theoretical Neuroscience at Columbia. The center is composed of 7 faculty members and 28 postdoc researchers and around 30 graduate students. We are a combination of physicists, mathematicians, computer scientists, and engineers.
The center was designed to be a highly interactive space where ideas could be freely shared among people with different backgrounds. Now, after the pandemic, we went from having a lively 3D interaction to a lively 2D interaction via Zoom. From the various and different collaborative studies that the center produces, today we are going to show you just a few. I am interested in the dynamics of cortical circuits. In the last years working together with Ken Miller, I focused on a specific microcircuit found across the entire cortex of the mouse brain. This microcircuit is composed of pyramidal cells and 3 different classes of interneurons, parvalbumin, somatostatin, and VIP. We collaborate with experimental laboratories, most of them focusing on the visual cortex of the mouse. Our collaborators are able to do simultaneous cell type specific calcium imaging recordings and cell type specific optogenetic perturbations. They have found that classic manipulations of the visual stimulus, that 2 of these interneurons engage in competitive interactions, the result of this competition directly regulating pyramidal cell activity in the visual cortex of the mouse. So to understand this better, my colleagues and I developed a battery of different complementary approaches to fit models to data. These fits result in mathematically tractable and data compatible encompassing models that describe the entire distributions of responses to stimulus. Due to their mathematical tractability, These models offer theoretical insights on the mechanisms underlying this competition and about the hypothetical outcome of specific perturbations and stimulus manipulations, and are therefore rich on experimentally testable predictions. These predictions can be carried out within the context of the collaboration.
I'm working at the intersection between deep learning and the brain. In the past couple of years, I've been developing the theory of object manifolds, which connects the geometrical properties of neural populations with the information content about object classes per neuron. This method can be used to characterize internal representations of deep networks and the neural data. So far, it's been used to understand how information untangles in visual, speech, language deep networks that are known to be similar to the brain. This is just the beginning of what can be done. A lot more is underway, from the neural data analysis to the theory of generalization in deep networks to measuring similarities between different tasks. In addition to developing theories and analysis tools, I've been also developing deep network models. These models are inspired by the latest findings in neuroscience, particularly by plasticity rules found in multi-layer structure in the brain. In a recent work on learning in electric fish by Larry Abbott and Nate Sattel lab, they found that signals useful for learning are very different from signals propagated for inference. Using this as a principle, Larry and I have been developing a network that consists of an ensemble of neurons that make inference of their own with their own local learning goals, unlike backpropagation in conventional deep networks. Last but not least, John Cunningham's lab has been building an exciting open source cloud processing infrastructure. This is a drag and drop cloud data analysis as a service like Amazon AWS. It's exciting because users don't have to install anything or know Python or CUDA, worry about library compatibilities, etc., but it provides an automatic full reproducibility with a much easier use and support. Because it's on the cloud, it has infinite scalability and it can run multiple data sets in parallel. If you're interested, definitely reach out to John to ask more. I hope many people will find their infrastructure useful. This is it, and thank you very much.
All right then. Thanks to everyone for such intriguing talks. And it's time now for questions. If you could please write your question in the chat room, and I will call on you. We have one question to start with from Julio, which is particular to Dr. Yoon. Julio, do you want to ask? It's about how far the light travels from the LED. And if you could show yourself on mute as you ask your question.
I think that you asked the question, and my question was how far did light travel from the LED and how much coverage you have in this column of activity that you are recording from, whether you have tested that.
It depends on the light intensity, but this micro LED can produce enough light to illuminate the vicinity of the lot of cells. But actually the purpose of this, the probe is illuminating putatively single cell, very small number of cells, so understand the local circuit connectivity. So we minimize the light down to the level that we can only illuminate or excite the neurons, or possibly single neurons, and monitoring the signal propagation. But if you can illuminate a huge power, maybe in the vicinity of the 100 microns and 200 microns, the neurons can be illuminated. But this is not an adequate the tool to illuminate the whole entire brain.
Thank you. Thank you.
We have a question from Vicky. Do you want to go ahead and ask it?
Sure. So this question was for Barani. And I was just wondering what the achievable imaging depth for the light sheet imaging is.
So yeah, this is a one photon light sheet. So whatever limitations are there for one photon is applicable to this particular technique also. That's why we are looking mostly at the insect circuits, which are not so thick. So we can easily do few hundred microns. But that's basically as far as what we have pushed so far.
And Bo, you had a question for Xiao-Jing?
Yes, I thought the results on spine density across areas was fascinating. And then there was something mentioned about the inhibitory connections. but I just lost what that result was. I was just wondering if you could repeat it.
Xiao-Jing, are you on?
Yeah, I was waiting.
And also, Julio can address that as well. So just to introduce, Xiao-Jing predicted, Julio then went and found substantiation. I'll let them talk.
So it was not really about the connectivity, it was just the proportion of different coverage cell types. Roughly speaking, if you just focus on two types, one controls the kind of target dendrites and controls the impulse to pyramidal cells, and the other one targets perisomatic region of pyramidal cells and controls the output. It turns out that the proportion or the ratio of the two type inhibitor neurons is not the same across areas. In the past, people tend to focus on PV neurons because they are predominant in sensory areas. That turns out to be not the case for higher areas. So the gradient is about the ratio of the two.
And maybe, Julio, do you know, would you have a prediction about the connectivity in that case?
So we don't. I think there's quite a bit of experimental measurements for the connectivity between those cell types in sensory areas like V1 or S1. But to my knowledge, there are fewer studies, of the same kind for frontal areas. So it would be really fantastic to be able to compare, at least, the two extremes.
Julio, did you want to add anything, given that's relevant to your published work?
No, I think that's Xiao-Jing actually made a good summary of that. I think that's
Very good. So, Tom, did you want to ask your question?
I'd like to know how many different LED colors can be included in the probes.
Currently, it is a blue, 470 nanometer for channel rhodopsin. But in other approach, we introduced the two different colors. One is blue, the other one is either yellow or the red for inhibition and also excitation. That's a waveguide approach. But currently, we're working on to introduce different color of LED for these probes.
Great.
And Kathy, did you have a question for SueYeon?
Yes, I was wondering if you could say a little bit more about the more biological learning rules that you were implementing in your deep neural networks and how they differ from traditional backpath approaches.
Yeah, so the network that we're currently building, so unlike the traditional DNNs where you have something that's equivalent to firing rate, this network has two signals for propagation. One is like regular firing rate, but with nonlinearity, there's an interpretation for probabilities for inference. So each neuron is has something like a probability distribution, and in turn that can be turned into a local loss function for each neuron. So you can write down the local learning objective function for each neuron. So it's somewhat like a probabilistic inference, but done in a very distributed way.
Can I ask a follow-up question?
Yeah,
sure. Are you implementing anything like sort of neuromodulator-based learning rules, such as have been described in like the insect mushroom body or something like that?
I think it has a connection to it because we have weights for the synaptic inference, sending the signal forward, but then they're selectively inhibited. So so it's context dependent and it plays a big role in the network. So it has a connection to it.
Is that it? So, Vicky, did you want to ask your question about training set size?
Sure. Yeah, it's a kind of a follow-up to Kathy's question too. SueYeon, have you had a chance to look at how the training set size requirements or training times compare using these more biologically realistic learning rules over traditional learning rules?
Maybe I'll just jump in here because it looks like SueYeon is so frozen in time. Yeah, we're definitely interested in that. I would say the biggest advantage we've seen is in non-destructive learning of multiple tasks. maybe more than what you're pointing out. But that's exactly the kind of things we're looking at. SueYeon, maybe you're back. Sorry, I jumped in because you were frozen.
Yeah, I'm back. Thanks for thanks for chiming in.
And anything? Did you want to add anything, or that was fine? Shall we go on to the next question? Okay.
Oh yeah, that's fine.
Okay. I couldn't tell if things were frozen or not. So Kristen, do you want to ask your question of Agostina?
Yeah, I'm not fully sure how to even ask the question, but you, at the end of your talk, talked about a kind of a database or a sharing tool that you indicated sounded like it might be faster and maybe accommodate more transfer of data. This is always an issue with serial section EM images, and so I was wondering if you might just tell us a little bit more. I think it was one of your collaborators is doing this work, but tell us a little bit more about that. And this was to Agostina and Larry's group.
Yeah, I am going to call in Taiga, who is the first author of that work. He's around.
Hello. Hi, everybody. I'm Taiga. Yes, I'd be happy to address that question. So we think of our tool more as an analysis sharing tool than a data sharing tool. But the idea now is that, let's say you have some sort of data set. We've worked with both, traditional, behavioral videos and also these calcium imaging, as well as other. We're thinking of working with EM sessions in the future as well. But now the idea would be that if you have some sort of analysis pipeline that you would like to run on it, then you'd be able to upload your data and then have automatically the results of that analysis pipeline. So, we're not really thinking too much in terms of the low-level data sharing aspect in terms of the efficiency, although that's definitely fast on the cloud. But, much more about this getting back the analysis without any of this extra effort. I hope that answers the question.
If you're still there, could you tell us approximately what's the size of data that's currently being, is it gigabytes, terabytes, or so?
Yeah, so far we've done experiments with up to 70 gigabytes, and we found that that's totally fine still. We have confidence that we can scale this further, especially if people want to go through like a programmatic interface. Yeah.
So I want to go back in the chat to a comment made by Tom Bartol. Would be interesting to explore variation of STP facilitation depression, so short-term plasticity across the brain areas. And I was hoping that multiple people might jump in to talk about that larger idea, because I think it's an exciting area for crossing between our NeuroNexs to explore these differences in brain regions and their really important but varied properties, such as short-term plasticity. Anybody want to jump in on that one?
I'd like to make a tangential comment, Amy, that may help. Can you hear me? It's Mark Ellisman.
We can hear you.
Yeah. So just to try and maintain my reputation as an iconoclast, there's a lot of emphasis on synaptic plasticity in all of this. And you mentioned in your intro these persistent firing interneurons.
They're pyramidal cells, actually. Well, some may be, some are probably interneurons, but the majority are likely pyramidal.
So one of the things that we know all the way back to Krajinovic is that neurons that fire frequently often have activity-dependent conduction velocity shifts. So in addition to the synaptic plasticity, the rate that the action potential goes from wherever that neuron is to its targets will vary depending on its history of firing. Now, this is not true in some systems because for timing circuits, you have to do something different. So I'm wondering if in these systems that you're looking at, which are really quite evolved, if there's any interest in understanding the axonal properties, which are also known to be quite plastic.
A lot of these are very short range connections. So Guillermo could speak to that, where he in vitro can actually see them in the slice going very short distances. We're focusing on the calcium in the spines,
yeah, I got that.
as a way of, but I'm not sure what changes there might be at the axon terminal in that way. Guillermo, do you have any comments related to that?
Yes, just a little comment. Something we studied in the monkey prefrontal cortex in vitro many years ago is actually short-term plasticity of some of the synaptic connections between neurons. And what I would say, I mean, we didn't measure conduction velocity and how this could change during an episode of repetitive stimuli that produce short-term plasticity. But from my recollection, if there were any changes in the latency of repetitive synaptic responses, it was kind of very small. So I would say, at least in our in vitro condition and for the particular synaptic connections that we studied, it looks like the conduction velocity doesn't show too much of a change.
Yeah. So it's a subtle property, but well studied in axonology. And what varies usually are the type of sodium channel and potassium channel at the nodes. It's not a terminal phenomena. There's something called the safety factor, where the action potential at, let's say, the first node in a series has enough current density to fire 3 nodes down the road. That's the way saltatory conduction actually works.
And does it have fewer KC and Q channels at those nodes?
For very fast, high-frequency tolerant nodes, as in the owl auditory system, you have 4.2 versus the 1 series, and they're located on the nodal membrane as opposed to the juxtaparanodal membrane. So fibers that do not allow, based on the animal's needs, conduction velocity shifts, do at least 2 things. One, they modify the potassium channel. They use a faster sodium channel. And then astrocytes that wick the potassium away from the nodes are more invested in those nodes. And that's what maintains the ability of the axons to have conduction. They fail at branch points. So if you have an arborizing axonal field, short or long, you'll fail to get to the terminal branches after repetitive activation, unless you have one of these adaptations. So I'm just saying that we're all still kind of in a synaptic LTP recovery phase as neuroscientists. But don't forget that axons are plastic, and that when you have persistent firing, you have to do something special.
Thank you, Mark. We will slide down the dendrite and see what we can see. Yeah, no, it's really important, especially with EM. You look based on your expectations often all too much. So it's important to be reminded.
Well, you look under, you're finding your keys under the streetlight, which is usually the synapse.
Exactly. In the interest of time, let's get to 2 other questions that are in the chat. Greg has one for Barani.
So I was interested in the imaging you were doing of second-order olfactory neurons, so mitral projection type neurons, and especially in the responses that you see in their axon terminals in the lateral horn. So you imaged both excitatory and inhibitory projection neurons, and I wonder if you saw differences in the temporal responses profiles of those 2 different populations. So one reason I wonder that is that they actually get different kinds of input on their dendrites in the antennal lobe.
So there is a short answer and there is a long answer to that. So in the interest of time, I'll keep this short. Yes, there are differences. The IPN responses are even more transient than EPN axonal responses. In fact, the EPN axonal response, the temporal response properties are quite different from the EPN dendritic response properties. So there is a transformation that happens between the input and the output compartments of the EPNs themselves. And there are differences in terms of both tuning as well as response profiles and what kinds of computations happen in the EPN axons and the IPN axons.
It's poster session for the long answer. Thank you.
And Jonathan, did you want to ask your question? about working memory? Jonathan Fritz?
Yeah, this is a naive question for XJ Wang and everyone else who's sort of been studying this, like Julio and others in this very exciting project that you're running, Amy. So as you know, at the beginning, I think Pat really had the idea that it was the spiking activity during the delay period, which was really important for working memory. And now it's clear there are a lot of other mechanisms involved. So I guess the naive question is, how are these different mechanisms integrated, not only within areas, but across areas? And I'm looking forward to reading XJ's latest paper, but in the theoretical modeling of this. But I think there's some interesting questions here about whether there's multiple mechanisms that are proceeding independently or whether they're really integrated. And that's the question.
Anybody in my group want to take that one on so I'm not doing all the talking?
I could start, and Xiao-Jing then can actually correct me if I know there is multiple mechanisms. There is memory, synaptic storage, calcium, and there is several oscillations and several things. One of the problems is that most of the working memory paradigms have been run with stationary displays. So you basically could afford that a synapse is not used until the delay period is up, and then the animal has to produce a response. So this is our emphasis in using naturalistic and virtual reality. In virtual reality, the whole brain is bombarded with information. The visual system is just processing a lot. Probably a lot of the synapses are getting used, or at least in visual cortex and in many other areas. And that's why we really want to probe those hypotheses using these naturalistic things. Because in reality, when you use working memory, you really have a lot of signals coming into the retina. It's very artificial to have a fixation point, a delay period when you have nothing on the screen, and then you have to produce a response. So it comes down to that. Truth, we really don't know. The evidence for persistent firing is strong though. But the other mechanisms, they need to be tested in more naturalistic conditions. That's what we think. I don't know, Xiao-Jing, what he thinks about that.
Maybe I'll just add that I think, excuse me, it's referring to the so-called activity silent state model or scenario. Maybe you can have some memory trace that's not reflected by persistent activity. I think that is a valid interesting candidate mechanism for a certain short-term memory trace. But I guess if you think about the definition of working memory in psychology, it not only involves maintenance, but also manipulation of information, right? So if you do need to manipulate information, internally, it's harder to think about how that can be done with passive processes, right? It's fully done through spikes. The other thing may be that, this mechanism especially is good for the latest thing you try to have a trace in your brain, but it will not be so resistant against the distractors, which is important characteristic of working memory, right? So maybe it helps to kind of differentiate working memory, kind of active, computation gated, function versus all kinds of short-term memory traces that just happen passively. I think that would help to kind of differentiate different mechanisms.
And I would add that they're also, of course, interactive, that you can have these short-term plastic events that support the persistent firing, so that it's not that it's one or the other. It's very often all of the above, interactions in these widespread networks where, for example, we know parietal cortex is engaged for spatial working memory. And I'm guessing that there are things like astrocytic calcium waves, things that we can't even measure right now, that are also having at least modulatory effects. So probably multiple things that interact to allow working memory. And we are at 10:55. And I see Guillermo has shared a relevant paper for Mark and others who are interested there on chat. But at this, I thank you all for your attention and your thoughtful questions, and we'll go on.